Bismillahirrahmanirrahim. 9700 A-Level Bio Paper 4-2 March 2024, and this is the first video. The paper is for 2 hours, as you can see, and it's a paper in which is very challenging, and you need to do the questions which you know well so that you can handle those questions and come back to the ones which you are not very sure of. Now, starting with question number 1, figure 1.1 is a diagram of a nephron. As you can see this diagram, then it says label figure 1.1 using one label line, letter A, to identify a region that contains urine. Which part would that be? It's the collecting duct. So A would be the collecting duct. Then one label line, letter B, to identify the region that contains podocytes. Now this is the inner part of the Bowman's capsule. So the inner part of the Bowman's capsule would be this part. So this would be B. And then it says C. One label line, letter C, to identify region of that is within the medulla of the kidney. Now you know the medulla of the kidney is here. It's all below this area. The whole of this is in the cortex. And then it's the medulla. So label line to the loop of Henle. This would be C, any part of the loop of Henle or part of the collecting duct below the level of the convoluted tubule. You could have even labeled C here. So loop of Henle or the collecting duct. Then the last one was D, identify the afferent arteriole. The afferent arteriole is the one which has a wider lumen. You see, this is all the blood going into the Bowman's, into the glomerulus, and then coming out of this. So this part would be the afferent arteriole, which is D. The afferent arteriole has a wider lumen. You can see this has a very small lumen. So afferent, efferent, exit. E for exit, E for efferent. Then coming on to the next part of the question. The B part of the question was a direct question, it's only for 4 marks. The cells of the proximal convoluted tubule are adapted to carry out selective reabsorption. Describe and explain how these cells are adapted to carry out selective reabsorption. Now you know they are something like this, and then you have the basal channels, and then you have another one here, another cell here, and the basal channels. Now why are they the basal channels? Because a lot of channel proteins, the sodium potassium pumps are here. So this increases the surface area for the sodium potassium pumps. And then of course you remember there is the tight junctions, which are the cell membranes are fused of these cells. So the microvilli are towards the lumen side, and the basal channels are on the basal side. This is where the basement membrane is present. The basement membrane is here on both sides. I'm not saying on this side only, it's on this side as well. So what you see is microvilli for large surface area, because the microvilli, these are the microvilli. Microvilli are the infoldings of the cell membrane, this side. So this is going to increase the surface area for reabsorption. Then transporter proteins, because you have special transporter proteins here, co-transporter proteins. So co-transporter proteins for the movement of glucose and amino acids with the sodium ions. Then the tight junctions here, this is the tight junction where the two cell membranes are fused, and this stops substances passing in between the cells or causes substance to pass through the cells. Everything can pass through this, but it can't pass in between the cells, and it should pass from one cell to the other. Then of course there are many mitochondria in it. If you just say mitochondria, that is not going to get you any marks. You've got to say many mitochondria. So many mitochondria to provide ATP for active transport. What do mitochondria do? They provide ATP for active transport. For active transport or the pumping of the sodium potassium pump works on ATP. Then folded basal membrane. This is the folded basal membrane. The basal membrane has a few foldings, but not like the microvilli, but that's called the basal channels. So folded basal membrane from many sodium potassium pumps. You can see here I showed that to you in the beginning. Many sodium potassium pumps in the basal membrane, because that's what's going to create a gradient. Sodium out, potassium in. 3 sodium out, 2 potassium in. So this is always going to be less sodium here When there's going to be less sodium here, this sodium here, which is in the glomerular filtrate, is going to diffuse in and it's going to bring glucose along with it. So this is how you have to understand it. I mean, I've explained it to you again, but these are the things that your very direct questions have come in many papers, and I'm sure everybody can give these 4 or 5. There are 5 points here in the mark scheme. So this is how we would write them. Microvilli for large surface area, co-transporter proteins for glucose, tight junctions to stop substances passing in between cells, many mitochondria to provide ATP, and folded basal membrane for many sodium potassium pumps. And you can look at this diagram and you can see for yourself how this is a very simple question and a very direct question. There's no ambiguity about it. Question 2. The scientist Gregor Mendel investigated differences in the length of the stem in the pea plant Pisum sativum. In 1866, he published the results of his investigation into the trait characteristics. Figure 2.1 shows a diagram of a pea plant. Then it says Mendel observed the pea plants he grew either had tall stems or dwarf or short stems. So there were only two variations, tall stems or short stems. In his investigation, Mendel carried out crosses using pea plants with these two phenotypes. So the phenotypes were only tall and short, and he carried out crosses. From the results of these crosses, Mendel demonstrated that tall stems were dominant to dwarf stems in pea plants. It is now known that the stem length trait in pea plants is controlled by one gene that has two alleles, a dominant allele big L small e and a recessive allele small l small e. Describe a crest that would be carried out and how the results of the cross could be analyzed to determine the genotype of a pea plant with a tall stem. Now they've told you that tall stems were dominant. So if they're dominant, they should either be this or they should be this. If it's a dominant trait, there are only two genotypes. If it's a recessive trait, then there is only one genotype. But here, of course, they told you. So it would be a capital L small e and a capital L small e. So either it will be this or it will be a capital L small e and a small l small e. Because there is a recessive. You see, just like I said, there's homozygous dominant or heterozygous. But here the letters used were big L and small e. So there's a difference. This would be a homozygous dominant, this would be a heterozygous. But heterozygous is not dominant or recessive, it's just heterozygous. Now, so how would you figure this out? Well, you do a test cross. Cross a tall with a dwarf. So if you crossed a tall with a dwarf, so the tall would be L small e, a big L small e, into small l small e and small l small e. Now all would be heterozygous. All would be heterozygous. So test cross with dwarf plant must be homozygous recessive. If all offsprings are tall, then the parent plant is homozygous dominant. If offsprings are 1:1, if any offsprings are dwarf, then the parent plant is heterozygous. Or you could have given me the breeding experiment, cross pollination, seed harvesting, then seed germination, then look at the phenotypes. You could have given me that detail as well. So as you can see, the answer was easy. Test cross tall plant with dwarf plant. Dwarf plant is homozygous recessive. If all offspring tall, then the parent plant is homozygous. So you see, if you do this into this, then all plants will be tall because this is the dominant trait. The tall is the dominant trait. Of course, they've told me to use this, but I'm just using this to make it clear to you all. So, I mean, by saying double capital A, I'm seeing it's actually, I mean, is big L small e, big L small e. But then if it's heterozygous where the ratio is 1:1, because remember this, when you do, it's very simple. When do you get it 1:1? When you do a heterozygous with a homozygous recessive. So you get a 1:1. 2:2 means 1:1. You simplify it. So this is how you do this question. B part of the question, the scientists P. W. Bryan and Hemming identified that the difference in the length of the stem in pea plants was associated with the presence or absence of gibberellin. They published their findings in 1955. Gibberellin leads to a response in plant cells by binding to specific receptor molecules. State the term used to describe a molecule as gibberellin that binds to specific receptor molecules and leads to a recessive. Well, that's a ligand. That's the new name in the new syllabus. Either you could have said ligand Or you could have said a hormone. You could have even said hormone, that was allowed. Or you could have said a cell signaling molecule. So you could have said any of these things. No, you weren't supposed to write there. So you could have said here ligand, which is simple, or a cell signaling molecule, or you could have said a hormone. Then 2b part 2 suggests the response of the cells in the internode region of the stem. You see, this is the stem, and then there is this branch coming out here and the branch coming out here. Now this is the area which is called the internode. Suggests the response of the cells in the internode region of the stem as labeled in figure 2.1 to the presence of gibberellin, and describe how this response affects the trait investigated by Mendel. So they've shown you here the internode. So in the diagram you can see the internode. Now they're asking you how the response affects the trait investigated by Mendel. I mean, what happens in the internode region for making it a tall stem? Very easy, cell elongation. Cell elongation, but you could have said increased cell division or mitosis, and then cell elongation occurs and the plant stem grows taller. Question number 3, cystic fibrosis is an autosomal. What is an autosomal? It's not on the gender. It's not on the 23rd pair of chromosomes, it's on the 22 chromosomes, which are the body chromosomes. So it's an autosomal recessive genetic disease. People with cystic fibrosis have a homozygous recessive genotype. So they have to be this. Now, if they have to be this, then the parents have to be this, because if the parents are normal, then they have to be this. That is how they can have a child with homozygous recessive. Now, the question they've asked you is, what is the term meant? Explain the meaning of the term homozygous and recessive. Homozygous means what? Both alleles of a gene are the same. Both alleles of a gene are the same. A gene has different alleles. Like I said, I have 5 colored markers. So I have markers, but they have 5 different colors. So homozygous means that both alleles of a gene are the same. What does recessive mean? Recessive is both the phenotype alleles is masked by a dominant allele. 2 copies of the recessive allele are needed for the phenotype to be displayed. But there are many ways you could have said that. You could have said must be homozygous for the recessive allele to affect the phenotype. The person must be this to be called recessive. It must have the recessive alleles to affect the phenotype. So you can word it differently. Effect of the recessive allele is masked by a dominant allele, or 2 copies of the recessive allele are needed for the phenotype to be displayed. Or you could have said alleles may affect the phenotype in the absence of a dominant allele. Or you could have said must be homozygous for the recessive allele to affect the phenotype. So wordings can be a little different, but you have to convey how you want to convey your message to the examiner. Okay, now question number B, part 1. In 2020, there were 10,800 people with cystic fibrosis in the UK. In the UK population was estimated to be 67, whatever, 1,000,000, 5 zeros. A proportion of people in the UK population are heterozygous for the gene. That means capital A, small a, that causes cystic fibrosis and do not have symptoms of the disease. So they are heterozygous that causes and do not have the symptom. They do not, they're not sufferers, but they have this heterozygous gene. Use the Hardy-Weinberg principle to calculate the number of people in the UK population who are expected to be heterozygous for the gene that causes cystic fibrosis. The equations of the Hardy-Weinberg are given. Now equation P plus Q is equal to 1, P square plus 2PQ is equal to 1, and all that. They've given you the whole information. This first stage of the calculation has been completed for you. 10,800 divided by the total. Now, how are we going to calculate Q? How are we going to calculate Q? Now, you've got to be really understanding how we're going to calculate Q. So, in order to find out Q, you just have the under root of that. So, Q is equal to what? 0.0127. Then it says what? You go to the next part of the question. P is equal to what? 1 minus 0.0127. P plus Q is equal to 1. So P is equal to 1 minus 0.0, or this is what? What is this? 0.987. So 1 minus this is the working, and 0.987 is the, now 2PQ is what? 2 into 0.9873 into 0.0127. Now this is what? If you just do this, this is 0.0251. Now how would you do the calculation for the heterozygous people? So this will be 67, whatever the population was, 1, 2, 3, 4, 5 into 0.0251. And this is what? This is 167 5,000, or you could have said 168 5,000. So this would get you your 3 marks. So you have to do a lot of maths, I'm sorry to say, but this is the maths that is expected of you in A levels. You could have also said, this was also a correct answer, if you said 168. This was also a correct answer. So you could have said this or this. Of course, if you had must be 3 significant figures or more, then the question comes right. Then part 2, the Hardy-Weinberg principle provides a useful estimate of the number of people in the UK who are heterozygous. However, the estimate is lower than the actual number. So the estimate is lower than the actual number. The underestimation occurs because not all the conditions of the Hardy-Weinberg principle apply. In the UK in 2020, the mean life expectancy of people with cystic fibrosis were approximately 50 years. All people were approximately 80 years. Explain how this information accounts for the underestimation of the number of people in the UK that are heterozygous for cystic fibrosis. Well, this is natural selection. Selection pressure is against the cystic fibrosis. People with cystic fibrosis have died. They do not survive as long as the normal people. And the heterozygotes are calculated from a smaller cystic fibrosis population. And some people with cystic fibrosis who have died will have passed on the cystic fibrosis allele to their children. So this was the reason why the estimates are wrong. The underestimation of the number of people in the UK, why that was wrong. So how do we word it? Natural selection, selection pressure against cystic fibrosis, people with CF do not survive long. Heterozygous calculated from a smaller CF population. CF is the word we specify that CF is, we're using the word CF for cystic fibrosis. So heterozygous are calculated from a smaller population. People with CF who have died will have passed on the CF allele to their offsprings. A screening program for cystic fibrosis was introduced in 2007 for all children born in the UK. Children are tested within 7 days of their birth. Children identified from the screening program as being at high risk of having cystic fibrosis can have a genetic test to confirm whether they have the disease. Table 3.1 shows the median predicted life expectancy of people born in the UK who have cystic fibrosis. Predictions are shown for people born in 2008, 12, 16 and 20. So year of the birth was 2008 and the median predicted life expectancy was 38.8 years. 2012, 43.5 years. You can see 2016, 47 years and 2020, 50 years. So it increased by about 12 years. Not much. Frankly speaking, not much. So the life expectancy increased. Now it says describe the trend shown in table 3.1 and outline how early screening for cystic fibrosis may have contributed to this trend. Early screening may have contributed to this trend. Now, what has actually happened? The predicted life expectancy increased over time. That's something when you describe, you must write that the median predicted life expectancy has increased over time. That's very easy. How has it increased over time? From 38.8, it has increased to 50.6. You got another mark for that. Increase of 11.8 or a percentage increase of 30.4. The reason is early screening will develop is treatment can be started early, and as better treatments have come in or improved care has come in between 2008 and 2020. So median life expectancy increased over time. Then you gave me the figures: 2008 30.8, to 2020 50.6, so increase of 11.8. Treatment can be started early. Better treatments have been maybe developed between 2008 and 2020. In many countries, a genetic test for cystic fibrosis is available for adults who do not have cystic fibrosis but have a family member who either has cystic fibrosis or is heterozygous for the gene that causes cystic fibrosis. And I usually, when I'm teaching this, I say, okay, well, if my brother's son has cystic fibrosis, that means what is my brother? My brother is heterozygous. Now, do I want to know whether I'm heterozygous or homozygous? Because if my brother has a child who has cystic fibrosis, then my brother is definitely heterozygous and his wife is also heterozygous. So that's why they have a child with cystic fibrosis. So in many countries, a genetic test for cystic fibrosis is available for adults who do not have cystic but have a family member with cystic or is heterozygous for the gene. These adults include partners, parents, offspring, brothers and sisters of the family member. The aim is to find out if any of these adults are heterozygous for the gene that causes cystic fibrosis. Discuss the ethical and social consideration of making a genetic test for cystic fibrosis available to these adults. Well, I don't know. I don't think I would like to have the test if I knew my brother is heterozygous. Well, if you do have the test, how is it going to help you? Well, it's going to reduce the worries if the result is negative. Then you can make an informed reproductive decision. Then the cost of the test, is it very expensive, or can the government do it, or is it a private test? Then you can plan for care of the child with cystic fibrosis. Then further genetic testing, you can do testing of the embryo, or you can do counseling. Then test is not 100% accurate. And then of course there is a problems. If you have the test, maybe there's discrimination, maybe there's problems in insurance, and then there's confidentiality. That do you want to tell everybody? Do you do not want to tell anybody? Are you okay with it? So all these things are social and ethical issues. So you can see reduces worry if negative, can make informed reproductive decisions. Cost of the test could be too much. Plan for the care of the child with cystic fibrosis, test not 100% accurate, problems of insurance and stigma. You see, social and ethical, whenever these questions are asked, the cost of the test, and then of course the ambiguity about it. Is it really a 100% okay test, or is it sort of not 100% accurate? That of course is a very major social and ethical consideration. Thank you very much. We'll continue the rest of the questions in the second video.